investigating anti-VEGF C and D therapy in wet AMD. Drivers of disease. In wet AMD, newly formed blood vessels grow abnormally in the choroid and retina, leaking fluids, lipids, and blood into the macula, causing tissue damage and rapid vision loss. This disease process is driven by the vascular endothelial growth factor, VEGF family of protein ligands, including VEGF-A, C, and D, that exhibit distinct binding specificities for the VEGF receptors 1, 2, and 3. VEGF-A, C, and D bind and activate VEGF receptor 2, the main receptor signaling for angiogenesis and vascular permeability, while VEGF-C and D are the only known ligands for VEGF receptor 3, which also is an important driver of angiogenesis. The wet AMD treatment landscape. Standard of care therapeutic agents for wet AMD primarily block VEGF-A. VEGF-A suppression has also been shown to upregulate the release of VEGF-C and D. Uncontrolled, these alternate angiogenic mediators may account for suboptimal visual outcomes in patients receiving current standard of care therapies. Although anti-VEGF-A monotherapy has been shown to stabilize disease progression and improve vision, it is not always sufficient in many patients to achieve or maintain significant improvement in visual acuity. Hence, a need exists for new treatments to improve the management of wet AMD. Combination therapy that targets alternative angiogenic factors such as VEGF-C or D may potentially provide additional benefit over current standard of care. Hope on the horizon. Introducing OPT-302, a first-in-class novel biologic therapeutic candidate specifically designed to trap and sequester VEGF-C and D. A recent Phase 2 clinical study, which enrolled patients with wet AMD who received intravitreal administration of OPT-302 combination therapy with ranibizumab, met the primary endpoint of demonstrating a statistically significant superior mean gain in visual acuity over ranibizumab monotherapy at week 24. OPT-302 combined with anti-VEGF-A therapy results in a comprehensive widespread blockade of ligands that activate VEGFR2 and 3 signaling in the eye, which may lead to broader inhibition of angiogenesis and vascular leakage compared with VEGF-A inhibition alone. In this clinical trial, OPT-302 combination therapy resulted in a similar safety profile to the ranibizumab monotherapy. OPT-302 is the only therapy to directly target the ligands VEGF-C and D. It is currently being investigated in 2 global pivotal Phase 3 clinical trials in combination with anti-VEGF-A treatment in patients with wet AMD. The primary efficacy endpoint of these Phase 3 studies is to evaluate if OPT-302 combination therapy displays superiority in visual acuity gains at 12 months compared to current standard of care treatments. With the potential to impact visual acuity, vision loss, and anatomical lesion measures, the future of treating wet AMD may be brighter with OPT-302. An innovation from Opthea.